Coagulase test, you have to know about the purpose of doing coagulase test and also the principle of this test. Coagulase test is a test used to differentiate Staphylococcus aureus from other coagulase negative staphylococci. What do you mean by that? So, Staphylococcus aureus is a kind of microorganism that can produce coagulase enzyme, while other staphylococci such as Staphylococcus epidermidis, Staphylococcus saprophyticus cannot produce coagulase enzyme. That's why they are coagulase negative. So this test is used to differentiate Staphylococcus aureus from other coagulase negative staphylococcus or staphylococci in general. Coagulase, as we say, is a kind of enzyme, a protein enzyme produced by several microorganisms, and this enzyme can convert fibrinogen to fibrin. That's why they will cause clotting. Coagulase is an enzyme. This enzyme can convert fibrinogen to fibrin by a process we'll talk about later. If you have a microorganism, and if the microorganism can clot plasma, it means this microorganism is coagulase positive. This microorganism can produce coagulase enzyme. As we said, coagulase can clot blood plasma by a mechanism which is similar to normal blood clotting. Coagulase producing bacteria, they are known as coagulase positive. As we say, they can produce coagulase enzyme. Coagulase enzyme can convert fibrinogen to fibrin and can cause clotting. What's the aim of doing this kind of fibrin formation or clotting? The aim of using this kind of clotting to protect the microorganism from engulfment. Normally, as a kind of our host defense, we have macrophage, and on the surface of macrophage we have receptors. These receptors can bind to several ligands on the surface of bacterial cells. So you have a bacterial cell here. This kind of bacterial cell can produce a coat of fibrin in order to protect itself from phagocytes. Okay, so this kind of fibrin formation will protect the microorganism from being engulfed by macrophage or phagocyte. We can say it's a kind of defense mechanism which can be used by microorganism in order to evade our immune system or our phagocytic system. The principle of this test, as we said, we have extracellular enzyme and we have a coagulase enzyme. This enzyme is known as free coagulase. Why? Because it's a kind of free enzyme outside of the body, outside of the bacterial cell, and is soluble. Free and soluble, same. Coagulase enzyme, so the principle of the clotting, you can see it from here. Coagulase enzyme will activate CRF. What do you mean by CRF? Coagulase reacting factor. Okay, so you have prothrombin in our plasma. Coagulase will activate CRF, and activated CRF will start in order to convert fibrinogen to fibrin, and after that the blood will clot. In order to see clotting and in order to get a part of blood without the cells, it's better to collect plasma, because inside plasma we have clotting factors. Same, you have principle here. This one is illustrated by a different method. You can see it here. We have fibrinogen inside our plasma, inside the plasma. So if you have fibrinogen, the blood will not clot. And if you have coagulase enzyme by the bacterial cell, it will start to convert fibrinogen to fibrin, and we said about that previously. and if you get fibrin, the plasma will clot. That's why in order to see coagulase test, in order to perform coagulase test, we need a substrate, and the substrate is plasma. So you have to prepare plasma, and after that you have to add the microorganism that you are going to test. Inoculation of the microorganism, add the bacterial sample to the plasma, and after that you have incubation. After incubation you'll see clot formation. If there is clotting, it means the coagulase positive. If there is no clotting, it means we have a coagulase negative bacterial sample. Staphylococcus aureus, beside the free coagulase, this kind of bacterial cell also has another kind of coagulase. It's known as bound coagulase. So normally regarding Staphylococcus aureus, we have two types of coagulase: bound coagulase and free coagulase. We said about free coagulase, and in order to test free coagulase, we need a tube method. We have to prepare plasma, and we have to put the plasma inside the test tube. After that, you have to inoculate the plasma by your bacterial sample. Incubation, and you have to check agglutination, or you have to check clotting here. Regarding the bound coagulase, it's also known as clumping factor, and you have to perform this test by using a slide. That's why it's known as slide coagulase test. So you have to prepare a slide. You can see it here. That's a slide, and you have to add plasma, and after that you have to add your bacterial sample, mixing and wait. Okay, in 5 to 10 seconds you'll see the result. If you see clumping or agglutination of bacterial cells, it means you have positive result. So this kind of microorganism has bound coagulase on the surface of the cell wall, bacterial cell wall of the bacterial cell. Okay, same, bound coagulase also can convert fibrinogen to fibrin. So the principle is the same, but here we have coagulase on the surface of the bacterial cell wall, but here we have free coagulase. Regarding bound coagulase, we need a slide method. Regarding free coagulase, we need a test tube method. As we said, as a substrate we need plasma. So how to prepare plasma, and what's the difference between plasma and serum? You know about the composition of the blood. Inside the blood we have different parts. We have cell, and also we have a liquid part. And if you collect the blood inside a test tube with anticoagulant, after centrifugation you'll get 3 layers. So this layer, we have a pocket red cell here, and here we have plasma the top, and this middle layer is known as buffy coat. But be careful, in order to see these 3 different layers you have to use a test tube with anticoagulant in order to prevent clotting. Regarding the types of anticoagulants, we have different types. Here we have EDTA, oxalate, heparin, and also citrate. All of them, they are anticoagulant. They will prevent clotting. In order to get plasma, you have to use a test tube with anticoagulant, one of them, citrate, EDTA, oxalate, or heparin. Rapid plasma also can be used, and this one is preferable. It gives a better clotting. It's free from inhibitors and is safe. Human plasma also can be used, but be careful, contains sodium citrate as anticoagulant. So be careful if you use a test tube with citrate, and if you have a contamination of some kind of bacterial cell outside of your original sample, you have contamination of Enterococcus faecalis, this kind of microorganism can utilize citrate, and if there is no citrate, the blood will clot and you'll get a false positive result. So be careful about this. You have some kind of contamination you may get a false positive result. The best choice is EDTA. Also, you can use oxalate and heparin. So it's better to think about that. It's better to use a test tube with EDTA or oxalate or heparin, and be careful, do not use a test tube with citrate. As we said, you may have some kind of microorganism, they can utilize the citrate. Normally, as we said, you have two methods: slide coagulase test and tube coagulase test. The purpose of doing slide coagulase test in order to detect bound coagulase, the purpose of doing tube coagulase test to examine the free coagulase. That's why we have test tube here, and here we have the slide. On the surface of the slide, you'll see agglutination, clumping of the bacterial cell. That's a positive result. And here you'll see clotting, and if you see clotting, that's a positive result. Regarding the incubation, the time of incubation, be careful. Regarding the slide method, you have to wait for about 5 to 10 seconds, but regarding the tube method, you have to incubate at 37 for about 4 hours, and you have to wait. If you see clotting after 2 hours, that's a positive result, but if there is no clotting, you have to wait for up to 4 hours, and you have to incubate the test tube at 37. Another explanation of the test, and you have to use your plasma here, and you have to inoculate your bacterial sample. Incubation, as we said, from 1 to 4 hours. If you see clumping, that's a positive result. If there is no clotting, if there is no clotting, you have to wait for 4 hours. If there is no clotting, after 4 hours, you have to take the test tube out from the incubator, and you have to put the test tube at room temperature. Why? We'll talk about the reason later. Here, same. We have plasma here, we have inoculation, and you have incubation. 3 different steps, or 3 main steps here. Bound coagulase, we said about that previously, that's a clumping factor, and absorb the fibrinogen from the plasma, and it will change the fibrinogen to fibrin, and it will cause a precipitation or agglutination or clumping of the bacterial cells. That's why you'll see agglutination. Tube coagulase test to detect both bound and free coagulase. Be careful about this point. So here in the test tube, you'll detect both of them, but regarding the slide method, you'll detect only bound coagulase. Some strain of Staphylococcus aureus may not produce bound coagulase. That's why we need a tube method in order to check, or in order to confirm the result. So tube method is better. Why? Because we can detect both bound coagulase and also free coagulase. Longer incubation at 37 may result in the disappearance of the clot. That's why we said after 4 hours you have to take the test tubes. If negative by end of 4 hours, incubate overnight at room temperature, and you have to re-examine at 24 hours or after 24 hours. Why? Because Staphylococcus aureus will start to produce staphylokinase, and it will disrupt the clot. That's why you have to incubate the test tube. After 4 hours, you have to incubate them at room temperature. Okay? The tube coagulase test should not be only agitated, as this can cause the clot to shrink and also giving you a false positive. So the clotting will start gradually. If you start shaking, you'll disrupt the clotting and you'll get a false negative. That's why just take it out and look at the clotting. If you see clotting, that's positive. If there is no clotting, do not shake. Okay, you have to return the test tube to the incubator, and you have to wait for about 4 hours. Red strains of Staphylococcus aureus are negative in coagulase test. Be careful. you may have a Staphylococcus aureus with a coagulase negative result. And also, besides Staphylococcus aureus, you may have other kind of bacterial sample that can produce coagulase enzyme, and they will coagulase positive. But regarding the genus of Staphylococci, if you get a negative result, it means you have coagulase negative Staphylococci.